Gene regulation in prokaryotes can be explained with the help of the lac operon model. Here the alteration in physiological and environmental conditions can be observed, leading to an alteration in expression in prokaryotes. It was observed by Jacob and Monner. The lac operon consists of regulatory gene I, it codes for the repressor protein. Z gene, it codes for beta-galactosidase which catalyzes the hydrolysis of lactose into glucose and galactose. Y gene, it codes for permease which regulates the lactose permeability in the cell. A gene, it codes for transacetylase which assists the enzyme beta-galactosidase. Hence, all these genes help in lactose metabolism. In lac operon, lactose acts as an inducer. If lactose is provided in the medium for the bacteria, the regulatory gene is activated. The inducer will bind to the repressor protein and render it inactive, which allows transcription of the operon. Thus, the lac operon is negatively regulated in this case. Gene regulation in eukaryotes. Gene regulation in eukaryotes is regulated by transcriptional activators and repressors. The repressors bind to specific DNA sequences and inhibit transcription. In eukaryotes, transcription involves several steps. It occurs in both nucleus, transcription, and cytoplasm, translation. Lac operon notes. Lac operon contains genes involved in metabolism. The genes are expressed only when lactose is present and glucose is absent. The operon is turned on and off in response to the glucose and lactose levels, metabolite activator protein and lac repressor. The lac repressor blocks the transcription of the operon. In the presence of lactose, it stops acting as a repressor. Metabolite activator protein activates the transcription of the operon only when glucose levels are low.